morning, everyone. Good afternoon, good evening, wherever you are listening to me. I am Professor Charles Oluwaseun Adetunji, a distinguished professor of microbiology, biotechnology, and nanotechnology. This morning, I'll be talking to us on application of nanotechnology for the production of agricultural bioinputs. As we all know, nanotechnology entails manipulation of matter at a nanoscale that ranges from 1 to 100 nanohem. Actual bioinputs, natural products that enhance plant growth and health, entails biofertilizer, biopesticides, and biostimulant. The need for integration of efficiency reduce environmental impact and promote sustainable agriculture. It has been stipulated that the whole world is going to increase drastically to 9 billion in the year 2015. Everywhere have now stipulated agricultural production we have to increase by 60% by then. Then agriculture should undergo a significant transformation that will help to meet the need of the ever-increasing population. This is the projection that is showing the trends how the increase in population is going to be in some years to come. There are four major goals of our time: achieving food security, avoiding dangerous climate change, health challenges, and high rate of unemployment. Actual bioinputs, these are biological derived substances or microorganisms used again to support plant growth and protect crop naturally. This can be categorized into biofertilizer, bioherbicides, and biopesticides, and biostimulants. Why do we need to integrate nanotechnology? Modern agriculture faces challenges such as overuse of synthetic chemicals and is causing a lot of soil degradation, soil dilapidation, and a lot of climate changes are being recorded. Nanotechnology now offers a lot of solutions that could help us, some of which entails efficiency in delivery of nutrients, targets. Why do we need to integrate nanotechnology? Why do we need to integrate nanotechnology? One, because of the efficiency of the nutrient, because of the targeted specificity, stability, and shelf life of our inputs, environmental sustainability by reducing chemical runoff and overuse. Now, let's talk generally how nanotechnology have come to stay in agriculture. The use of nanoscale material and technology in agriculture is very important for a lot of agricultural processes. There are some key tools that we need to understand. One of them is nanoparticle for delivery of nutrients and agrochemicals. Another one is nanosensor for real-time monitoring of soil, water, and crop condition. Then nanocarrier for control release system of fertilizer, pesticides, and bioinputs. What are the benefits? It increases precision in input application, enhance efficiency of nutrients and pesticide use, improve environmental sustainability by reducing waste and pollution. Some of them include application of the right amount of input at the right time in terms of precision, minimize input is used, efficiency, I've said that, and sustainability lowers environmental impact and reduce reliance on synthetic chemical. Type of agricultural bioinputs. We have biofertilizer, which are organisms that enhance nutrient availability such as rhizobium and azobacter, phosphorus-solubilizing bacteria. We have biopesticides derived from microbes, plants, natural substances to control pests. We have biostimulants and bioherbicides, biofungicides that help to suppress fungal disease like trichoderma, azanium, and Pseudomonas fluorescens have such activity. Now, nanoagrochemicals. They are the key drivers for applying nanotechnology to improve efficiency of agrochemicals associated with socio-economic and environmental consideration that still need to be addressed and are shown in a round table. Now, our group have done a lot of work extensively on nanochemicals. Why? Because it's more efficient in terms of application, because of stability, suspension, homogeneous coverage, more precise target area, lower application, reduce losses, reduce photolysis and transport to non-targeted area, improve interaction such as improve uptake, control release, targeted delivery. Now, This has made nano-agrochemical very essential in order to ensure a stable food and increase in agricultural production. Our group have produced 6 good books on nanotechnology and nanobiosensor, and you can see them, you can get them to your library. Now, biofertilizer are living organisms that promote plant growth by increasing availability and uptake of no primary nutrients. Examples include nitrogen fixers like rhizobium and azobacter, which convert atmospheric nitrogen into a usable form. Phosphate solubilizing bacteria that convert soluble phosphate into a form of accessible plants. We have also some other we have biopesticides. Biopesticides are environmental friendly alternatives that could help. Biopesticides are environmental friendly alternatives to chemical pesticides. They can come in form of microbial sources such as bacterial centroidensis, which produce protein toxic to insect larvae. We have botanical sources like neem for its broad spectrum and insecticidal activity. Biochemical agents include pheromones of plant-based toxins that interfere with pest reduction and feeding. Biostimulants are inputs to improve plant vigor, yield, tolerance, and abiotic stress like drought and salinity. Example includes agar extract, amino acid, beneficial microbe like isospirillum that stimulates root growth. Herbicides are biologically based weed control agents that are usually involve pathogenic fungi or bacteria that selectively infect weed species. Example include Clostridium geopodis, Alternaria casei. Our research group have developed a new herbicide that can assist farmers to prevent the usage of synthetic herbicide, which is one of the threats to food safety and ecosystem and human health. Now, during this research, we were able to isolate and apply cultural, molecular, and a lot of other techniques in order to isolate the best strains that could produce this bioherbicide activity. We have established the isolation and purification of DNA from such isolate. We have performed PCR reaction method and it was verified on Agorist Joy. We also perform intertarsal spacer D1 D2 region of the sequence of the Lasovarian syndrome domain, and also this showing the geographical summary alignments to show the alignment of the sequence producing significant alignment with the Lasovarian pseudotrombane. This is showing the distance obtained from the neighbor joining analysis from the interstitial spacer, indicating the phylogenetic relationship among the isolate and the particular species. This is showing the publication we have published from after isolation, identification, and characterization screening of the rhizophoric bacteria for their herbicidal activity. We also went further to establish the metabolimic profiling by isolating and establish the structural isolation and biocidal activity from this particular strain. We determine the mode of action on the weed and the non-target as well as electron scanning microscopy studies. This is showing the publication from our research group based on the influence of the eco-friendly phytotoxic metabolite from Lasiviridia pseudotrombane on the physiological, biochemical, and cellular structures of the weeds. We went further to establish this on using scanning electron microscopy, and this showing the activity. You can see the control weed, the treated weeds, and showing that they could be infected after application of this bacteria, so that we show and establish the biocidal. This is showing the award given to me after the completion of the work on bioherbicide. So this is the publication documented from that research. And we went further to perform the metabolomic profiling of the bioherbicidal strains. And this showed from 2 liters scaling up to a bioreactor of 1,000 liters, and the purification and characterization of the active compound. We went, and this is showing the structural isolation of the compounds responsible for the bioherbicidal activity. We went further to establish structural isolation of the active compound using HPLC, MS, FTIR, SCMS, and proton and carbon NMR. And we went further to establish the using carbon phototoxicity, which were determined by the tolerance enhancement polarization transfer using depth 45, 90, 135. COSY were also performed using bronchial microgram. So this is the publication that established all the structural compounds. We went further to establish the herbicidal and enzymatic activity of, as well as screen the economically viable substrates available agricultural waste for the mass production of these strains. And this is another publication, and this is the publication we were able to publish from the use of agro waste production with sustainable efficiency. We went further to establish which one is the best, whether solid state fermentation, submerged fermentation, or biphasic fermentation. And this is the publication establishing uh, uh, submerged fermentation, and this is the picture showing solid state fermentation. And we went further to establish the ecotoxicological assessment of the bioherbicide. In order to establish the biocompatibility of the bioherbicide on the health status of the soil and mechanical responsible recycling of the essential nutrients like nitrogen, carbon, sulfur, and phosphorus, we also perform the following activity like CO2 evolution, carbon dioxide evolution, organic soil content, enzymatic activity like acidic and alkaline phosphatase, the hydrogenase, urease, and protease, and the population responsibility of each soil microorganism. And this showing the scheme of work that was performed in an in vitro trial by herbicidal formulation, and we use glycophosphate as a positive control, and we produce a pesticides without any organism plus an adjuvant serving as a negative control. And this is showing the scheme of work that was performed in order to establish the effect of the bioherbicidal activity on soil organism, soil activity, soil carbon, and we later establish a lot of bioherbicidal acid. This is showing the publication we eventually published after establishing the hypothesis and confirming the activity of such bioherbicidal strains. We went further to carry on farmer because we believe so much in translational research. So this is showing the farmer that we performed the experiment together in order to establish the activity of such. We also carry agric economists, extensionists along in order to validate the economic value and the adaptability, acceptability of such a bioherbicidal strain. We have also documented a patent in this regard. Now, let me talk about biofungicide after I have elaborated so much on bioherbicide. Biofungicide are naturally occurring compounds or substances that could prevent fungi in crop and disease, that prevent fungal disease in crops. They work through competition, parasitism, and induce plant resistance. Common agents include trichoderma and bacteria such as Pseudomonas fluorescens. Now, our research group were able to also make use of cellular contents and micromolecules, or whether small molecule or micromolecules from these organisms. And one of such is a new raminology that was detected to be a safe biosurfactant with a diverse and unique property. It could be a permanent replacement to costly, environmental, non-friendly, earthly, unfriendly synthetic chemical used in the management of plant pathogen, in biological control of medical significant pathogen also. And this is showing the picture that I was able to take after the completion of projects of this biosurfactant. And this is showing the from fermentation to purification, standardization, optimization, and activity of such raminolipids. We went further to optimize the effect of the mycelium on the best pH, temperature, and a lot of other conditions to establish the best biological activity. We went further to publish the activity on characterization, optimization of such raminolipids with novel biosurfactant activity. We went further to establish the enzymatic activity of such compounds, raminolipids, on pathogens. We also went further to establish the effect of synthesizing nanoparticle with the metabolite of such fungicidal bacteria. And we have also documented a patent in such regards. And these are the 5 textbooks on surfactants we have been able to also publish. So nanotechnology in biofertilizer production is very effective because of nanoencapsulation of the microbial inoculation. One major challenge of using biofertilizer is survival of the beneficial microorganisms in storage and after soil application. So nanoencapsulation using nanomaterials like tituzan and silica film, protective coating around microorganisms like rhizobium and aspergillum, and they shed it from UV light desiccation and temperature extremeness. Also, encapsulation allows gradual degradation and release, targeted and controlled release of the active compound over time. So control release of nutrient is very important. Nanotechnology enables precision delivery. For instance, nutrients like nitrogen or phosphorus can be bound with nanocarrier that release their content slowly and steadily. So this reduces nutrient loss and through leaching of so plants receive more nutrient more consistently, improving uptake and reduce need for repeated application. Improved microbial viability and soil colonization. Nanotechnology can help to enhance adhesion to plant roots and promote rhizosphere colonization, increase efficacy of nutrients and fix or solubilizing process. Emulsion for better solubility and stability. Biopesticides, especially those derived from plant and microbes metabolites, offers is also a solution for poor water solubility and instant under the environmental condition. Nanoemulsion and water in oil or water in oil system that has a droplet size in the nanometer range, which increase the expansion of active ingredients. This formulation of higher surface and improved penetration and longer shelf life. So example is neem oil nanoemulsion are more effective and lower and require lower dose when compared to another one. Targeted delivery to pest sites using nano carrier is also very important because targeted delivery to pest sites using nanoscale ensure bioavailability of the active ingredient at the active site. Targeted delivery minimize loss of environmental degradation, improve overall pest control and effectiveness, and allow response release where the active compound is released in response to specific triggers like temperature, pH, and enzymes. Reduced toxicity in the environment is another one because nanotechnology formulation allow lower dosage and reduce non-target effect. So control release system prevents excessive application and help in sustainable pest management. What are the advantages of nano bio inputs? Higher efficiency in terms of lower dose, they are readily absorbed by plant and microbes, and farmer can use them in smaller amounts while achieving equal or better result when compared to the traditional formulation. Extended shelf life of bio input because nano encapsulation and nano carrier protects biological material from heat, moisture, and oxidation. In fact, they extend the viability and effectiveness of bio-input over a longer period of time, reducing losses. Protection from UV degradation and environmental factor. Nano carrier made material like chitosan silica acts as protective barrier for shielding maintenance and activity and potency of bio-inputs in feed, especially under harsh condition. Precision targeting and reducing waste because they minimize runoff, lynchings, and fluorization, leading to less environmental contamination. There are some limitations concerned on the use of nano bio-input that a lot of people have raised, like regulation concern because many countries lack clear guidance for evaluating safety, efficacy, environmental impact of nano materials in agriculture. So absence of standardization in terms of toxicity testing protocol and registration processes create uncertainty for manufacturers and users. Potential toxic to non-target organism because some soil microbial are essential for certain nutrient pollutants like B, a lot of other things. So this needs to be taken into consideration. Cost and scalability is very important for resource poor farmer, especially in developing countries. Having access to this is limited. Scaling production to meet global demand also present logistical and economical challenges that needs to be focused attention on. Need for long-term environmental studies. Most current studies are short-term and lab-based, focusing on immediate present plant response to toxicity. So there is a need for a lot of field trial in different ecological zones in order to be able to establish which is the best treatment. And also there is a knowledge gap in understanding how particles behave in a refeed condition over time. The question remains about the accumulation in the soil and potential for accumulation in the food chain and impact on biodiversity. So current research and case study. Notable research and real-world application of nano inputs have been performed, like nano trial, nano fertilizer fire. One of the prominent examples is the use of nano rhizobium, a nano formulation fashion of nitrogen fixing bacterium using rhizobium. Research have shown that encapsulation of rhizobium in nanoparticle like silica or chitosan can enable microbial stability during storage and application, enhance soil colonization and root adhesion, better nitrogen fixation, which has led to improved plant growth and yield. Study have also explored other microbial formulations such as nano-encapsulated Aspergillum fusarium group. Nanochitosan is also apply as a biopesticide because chitosan is the kind of biopolymer derived from chitin, the exoskeleton of crustacean, and has a lot of antifungal and insect repelling property. Research have shown that nanochitosan increase effectiveness of biopesticide like enhancing stability and penetration in plant tissue, improve pest control against various insecticides and the pests, reduce need for chemical pesticide integrated pest management system. Nanochitosan has been successfully tested on crops like tomato, pepper, cucumber for controlling fungal infection and a lot of other aphids. A lot of institutions around the globe have performed very wonderful research. In Nigeria, my research group is doing a lot of things on all these aforementioned nanofungicide, nanopesticide, nanoinsecticides, nanopesticides. Also in India, the research institutes such as India Institute of Technology and other research institutes are being on extensive research on nanobar inputs for sustainable agriculture. So research on nanofertilizer, nanopesticides, and nanofertilizer is a significant focus that we need to focus attention if we are to achieve food security and food safety. United States, like UC David and Cornell University, are conducting experiments on nanofertilizer, fertilizer, and plant growth enhancer. European Union has funded a lot of projects and gave a lot of grants like for projects promotion like nano agri, nano farm, even at improving food security and sustainability through nanotechnology. Chinese university and cultural research are leading in nano-based pest management and fertilizer technology for enhanced nutrient delivery and crop management. So, if we are to achieve this, we have to focus more on a lot of other future prospects. The features of the nano bio input in agriculture is very sure. Like if we can embrace smart nano formulation with AI-driven delivery, integration with precision agriculture, because use of drones, satellite imaging for real-time monitoring of crop health, nutrient level and pest activity, GPS guided system and soil sensor is going to work. Also, incorporation of policy and industrial collaboration scale for nano input to be widely adapted. Collaboration needs to exist between agricultural industry, research policy maker. Government needs to establish clear regulation framework and safety standard to ensure safe use of nanotechnology in agriculture. Public-private partnership will help us to scale production and ensure this technology are accessible to both larger and small-scale farmers. Industry collaboration can drive innovation to create a cost-effective solution that are more scalable, easy to use on a global scale, making nano input viable for wide application, even in resources limited area. In conclusion, today's talk have established a scientific community and enlightened us on what nanotechnology could offer as a significant advantage in agriculture, particularly in developing nano bio inputs like higher efficiency, environmental sustainability, precision agriculture, and some of the limited challenges and regulatory frameworks concern environmental risk, scalability and cost, need for long-term research, and we need to more focus attention on the application of nano to revolutionize sustainable agriculture. Despite challenges, nano holds immense potential to transform agriculture by making it more sustainable and efficient. Nano by input could revolutionize the way we grow food, protect the environment and human health by minimizing the use of chemicals like fertilizer and pesticide. Research and innovation continue. We expect a smarter and a more application that will make farming more resource efficient, climate resilient, paving way for a more sustainable agricultural future. On this note, I would like to appreciate Professor Luciano Provano Silva, the chair and organizing committee of the 6th International Conference. You can get more of this international talk and scientific talk on land application of nano in terms of bio input in agriculture in my inaugural lecture. Thank you very much. I would like to appreciate a lot of country and a lot of other people that have supported my research work. I'm very grateful. Let's keep in touch and continue to do a good research. Until we meet on the next episode of this conference in 2026, I say bye-bye for now and continue to do good science. Bye-bye.